I'm Susan Denser, a member of the Dartmouth class of 1977 and Senior Policy Fellow at the Robert J. Margolis Center for Health Policy at Duke University, based here in Washington, D.C. I'm also a former member of the Dartmouth Board of Trustees and was a longtime member of the Board of Advisors for the Geisel School of Medicine. I'm really honored to be moderating today's program. Well, as we all know, fighting a pandemic requires an arsenal of medical countermeasures. Those are life-saving medicines and medical supplies that can be used to diagnose, prevent, protect, and treat conditions associated with infectious diseases such as COVID-19. And as we also know, COVID-19 vaccines have now been developed, tested, manufactured, and approved in record time. Several types of therapeutic antibodies have also been approved for use by the U.S. Food and Drug Administration. And meanwhile, with respect to vaccines, though, the nation is encountering difficulties in delivering them over what's known as the last mile and getting shots out of the vials in which the vaccines come and into human arms. All of that has proved challenging. So what's worked well and what hasn't worked so well in terms of translating basic scientific discoveries into effective countermeasures and then getting them into broad use in the real world? Our distinguished panelists today are going to dig into these questions, and we'll take up others that many of you will pose today for us as well. Let me introduce our panelists briefly now. First with us is Kendall Hoyt. She's assistant professor of medicine at the Geisel School of Medicine and a lecturer. Oops, sorry about that. I'm just, something just happened with my notes, so let me pull those quickly up. So Kendall, as I was saying earlier, is an assistant professor at the Geisel School. She's also a lecturer at the Thayer School of Engineering, where she works on technology and biosecurity. Kendall has also worked with the Coalition for Epidemic Preparedness Innovations, CEPI, which is a global partnership developing vaccines to stop future epidemics. And we're going to hear more about her work with CEPI very shortly. We're also very happy to be joined by Tilman Gerngross, who's professor of bioengineering at the Thayer School of Engineering. He's also adjunct professor in the departments of biology and chemistry at Dartmouth. He's been an active inventor, and to date, his work has resulted in the founding of 5 companies, including Adimab, which is the global leader in therapeutic discovery and engineering of human monoclonal antibodies. Adimab happens to be based near Dartmouth in Lebanon, New Hampshire, and Tilman holds more than a dozen US and international patents. And we're very happy also to be joined by Jake Reeder, who's the co-founder, president, and CEO of Celdara Medical, which is a company that transforms academic innovations into medicines for the world's most challenging diseases. Jake and his team at Celdara also formed the Pandemic Security Initiative. That's designed to protect the nation from future pandemics by developing drugs that integrate the best of groundbreaking science, entrepreneurial innovation, public sector investment, and private sector efficiency. They started that work in 2014. Last year, they pivoted to assist in the response to COVID-19. Before he founded Celdara, Jake founded the New Ventures office at Geisel, where he still serves as director. We were going to be joined today by Daniel Rapp, a graduate student at the Guarini School of Graduate and Advanced Studies. Unfortunately, he is in Texas today and is without power and without internet. So sadly, Daniel will not be joining us. We'll make the best of it with this distinguished panel that we have, I'm sure. Before I get started, I want to run through a couple of housekeeping items. First, we want this to be very interactive, and we'll do our best to respond to as many questions as possible. So please submit those through the Q&A button, which is at the bottom of your webinar window. If you see a question that you're interested in, please upvote that question to make sure it rises to the top. Please use the chat box for any technical challenges you might experience during today's program. This webinar is being recorded and will be made available to all attendees after today's session. And finally, you'll be receiving a survey at the end of the program. Please take a brief moment or two to provide your feedback. That helps us enhance future programming. So to kick off today's discussion, I want to start with a poll for all of you in the audience. So let's bring up that poll now. And we're asking here, as you see, are you concerned about how quickly the COVID-19 vaccines in particular were developed and tested? And you can see you've got three choices. You're either very concerned, somewhat concerned, or not concerned at all. So let's go ahead and take that vote now and see where the audience comes out on that. Okay, so no, not concerned is basically at least more than half of you are not that concerned, but a significant people are a portion of people, I should say, are either somewhat concerned or very concerned. So that's a good starting point for our discussion, and we can help now to elucidate what are the grounds for concern or not about the rapid pace of development of the vaccines. So turning to our discussion, Kendall, we're going to start with you and your work with CEPI in recent years to prepare for pandemics and the vaccines that we hope will be able to help us all defeat them. This role at CEPI, and of course your work at Dartmouth also, has given you a front row seat in the development of these vaccines. And you've got a lot of perspectives on how this work has gone, but also how it could have gone better. So give us your overview. How do you think the world has done approaching this pandemic through the lens of vaccines?
Thank you, Susan. I think it's important to realize just how extraordinary it is to have safe and effective vaccines at this stage. I mean, the US authorized 2 mRNA vaccines approximately 10 months after China posted the first sequence. And vaccines can take 10 years when you're starting from scratch. And 90% of them can fail. So 1,000 things had to go right for this to happen, and they did. So I think this is a moment for even the staunchest skeptics to dance in the streets. But I mean, there is more work to be done to keep up with this virus and to prepare for the next one. So first, what did we do right? Number one, we did not start from scratch. Previous work on SARS and MERS, both coronaviruses, taught us a lot about the structure and the importance of the spike protein as a vaccine target. Researchers had also been working on the mRNA platforms, in part because they recognized their value in an outbreak. So once you know the sequence of the viral protein you want to target, these vaccines are relatively easy to make. So it's not surprising that they were the first across the finish line. That we also had high rates of transmission, which is not a good thing, but it allowed us to test vaccines very quickly. Operation Warp Speed facilitated funding, coordination, manufacturing, supply chain management, all of that mattered. The FDA was incredibly responsive and flexible, and they deserve a great deal of credit. So Susan, you mentioned CEPI, which plays an important role. They are a nonprofit pipeline accelerator that funds the development of vaccines for diseases with pandemic potential. And the market often fails for these vaccines, in part because companies cannot respond to demand, which is characterized by these unpredictable flares. Think of SARS, Ebola, Zika. They exploded and then they disappeared and there was no market. COVID generates global demand, but this also creates problems, which we'll discuss. So I helped write the business plan for CEPI, which launched in 2017, and led the response strategy team. So CEPI uses a two-part just-in-case and just-in-time strategy. One, they fund a handful of high-priority pathogens to phase 2B, just in case there is an outbreak. It might not be, they might not have made the right choice, but then there's something to roll out for testing just in case. This really was the lesson for Ebola. Two, they invest in platform technologies like mRNA or viral vector vaccines that can respond to a wide variety of pathogens quickly. In preparation for the unknown, CEPI also builds flexibility into their contracts with developers. So as early as January of last year, CEPI leveraged both their MERS contracts, pivoted on their MERS contracts, leveraged on their platform development technologies to have 9 COVID vaccine contracts. So the R&D is moving quickly, but But this next phase has been problematic. Just 2.3% of the Operation Warp Speed budget went to distribution. So you can have the best vaccine in the world, but it doesn't do any good if you cannot get it into people's arms. And this is a really urgent problem, not just in the US, but globally, even more so globally. We have to stop this virus from replicating and mutating on such a grand scale. We're seeing variants, they're exploding everywhere. It's a natural consequence of this rate of transmission. So last spring, CEPI got together with Gavi and the WHO to form something called COVAX, which is a mechanism to procure vaccine and ensure global access. Problem is, while they were trying to raise money for this, rich countries bought up supply, which drove up the price and undercut efforts to distribute on the basis of need as opposed to the ability to pay. And this runs the risk of prolonging the pandemic because you want to end chains of transmission as efficiently as possible. So what did we learn? We learned, number one, we cannot pass the hat for funding in the middle of a pandemic. We need a sustainable resource mobilization strategy. We need to sustain and build accelerators like CEPI and platforms like COVAX that will allow us to produce vaccines as a public good, not as a commodity with speed, scale, and access. So.
Great. Well, thanks, Kendall. And you've obviously made the very important point that we need an entire pipeline to bring these countermeasures to the public, starting with the basic science, but all the way through creating the appropriate market conditions and pulling everything forward into the actual use in the public realm. So thank you for making that point. And to the point that you were starting to get to about equity, I just want to remind everybody that the third and final webinar in this series on February 22nd will deal on that topic. It is called Eye on Equity, COVID-19 vaccine distribution in the US and worldwide. So I hope everybody on this call today will join us for that one as well. So Tillman, let's go to you next. In your very extensive work, turning scientific discoveries into effective therapies, you, as I mentioned, founded Adimab. That company recently spun off another entity developing a portfolio of anti-coronavirus antibodies as both therapeutics, as I understand it, and also prophylactics against COVID-19 and future coronaviruses. And we know that there are many out there. So you're right in the thick of this surge of discovery and development activity around SARS-CoV-2. Give us your perspective on what's occurred to date. And if you would, tell us about this very interesting contrast that is developing now between vaccines and antibodies. Vaccines right now, we have far more demand than we have supply. With antibodies at the moment, it looks like we have more supply than we have demand. I hope you'll explain for us why that is.
Absolutely. So let's break it down. I don't remember all your questions, Susan, so please remind me as we go through it. Let me perhaps share some slides with you to describe to you of how we got here and how we perceive the situation, because everyone that was approaching this emerging pandemic, thought about it differently, approached it differently, and ultimately deployed different strategies. So I'm going to share with you just a few slides on that very topic. Oops, sorry. Let's try that one more time. A few slides, there we go. So we currently are in a situation where the entire world is talking about coronaviruses, in particular about SARS-CoV-2. And what the current focus on the current outbreak misses is something that Kendall alluded to and Jake has worked on. So I think it's a commonly accepted understanding in the scientific community that coronaviruses have been a significant threat to humanity for some period of time now. And in particular, the family here, the beta coronaviruses, have led to multiple endemic viruses, OC43, HKU1. These are viruses that cause mild respiratory disease, cold-like symptoms, but are endemic in the human population on essentially every continent. This family has led to the MERS outbreak in 2007, 2008. And in particular, the current outbreak is a particular lineage B of the beta coronaviruses that target a particular human receptor that we all carry called ACE2. And so there's a whole class of viruses that include SARS, SARS-CoV-2, that exploit this particular receptor in human airway epithelial cells to get into cells and then proliferate. And so we've seen an outbreak in 2002, 2003 in the form of SARS, and we have now seen an outbreak 2019, 2021 with SARS-CoV-2. And it is this class that I would be particularly concerned about because we know a lot about it. Not because work I did or anyone at Dartmouth did, but the scientific community, in particular 2 labs, one in the US, Ralph Barrick's lab at UNC, and Shengli Shi at the Wuhan Institute, they've been studying these viruses for many, many years. These are folks that go out in southern China, they put up nets, they catch bats, and then they characterize what viral infections are present in those bats. And the emerging picture is very scary. Anywhere between 6.5% and 25% of bats in southern China carry coronaviruses. Many more than one. So one bat may have multiple infections at the same time, and then they all go back into their bat cage and they hang out together, creating what we now call a super spreader event. And this is the animal reservoir in which recombination of these viruses occurs, and then on occasion they spill over into humans. So if you look at the human population in southern China, about 1 to 3% of people living in that part of China show serological evidence of coronavirus infections. This is long before we got into this situation. So we have an animal reservoir where recombination occurs, and then it spills over into humans, and often nothing happens. But a few mutations, like in our case here with SARS-CoV-2, the introduction of a protease cleavage site makes the virus more fit. And all of a sudden it spreads into the entire world. So we've seen this with SARS, same origin, SARS-CoV-2, same origin. And it's horrifying because you read the scientific literature and like these early papers from 2016 describe this virus WIV1, and Ralph Barrick's group in this paper talks about how this virus that targets human ACE2 is a warning of possible reemergence of coronavirus outbreaks. 2016, a mere 3 years before we had a global pandemic. So let's start by saying anyone who says, who would have thought, just hasn't read the literature. This was all out there in front of our eyes. And we're now, because we're in the middle of a pandemic, paying real attention to it. And so it is our view, and this is how we approach this differently than everyone else, we need a solution to this class of viruses that target human ACE2, because that protects us now, that will protect us from variants that are currently emerging, and that will protect us in the future if another family member of this particular class of viruses called sarbecoviruses spills over into humans. And there's not a question in my mind of whether it will happen, it's a question of when will it happen. So that is how we took a very different angle of how to solve this particular problem. So I'm not sure if I answered any of your questions, but I felt it was important for the audience to understand that this is a looming problem that's going on while we're talking on this Zoom call here. There are bats hanging out in China that are recombining coronaviruses that may very well lead to the next outbreak down the road. So anyway, that's the observation I wanted to share.
That is terrific, Tillman. And we'll circle back and ask you more about therapeutic antibodies as a countermeasure, but also very particularly, and Jake, I hope will address this too. So if we know that this is coming, that this is out there, how do we get ready? How are we in an even better position for the next pandemic? So Jake, that leads us directly to you. Could you talk about the pivot that I described earlier that Sildara took around the pandemic and build on some of the recommendations you've already heard about how we position ourselves better for the next pandemic, as well as we did at least on vaccines, as Kendall pointed out, we have to be in even better shape for the next time.
Yeah, thanks, Susan. Fantastic panel. I've really enjoyed the first two panelists. It's an honor and a pleasure to be here talking about what is really an existential topic here. I want to first start by picking up on something that Kendall said and start with a thought experiment. So if we imagine that we had a perfect cure for COVID-19 in 2015, say, what would that have looked like? We would have spent a few $100 million developing the drug, and we would have had $0 in sales, right? And so that's the first problem, right? Free market capitalism can be a wonderful thing when there's a market. But as Kendall was saying, there is no market for sporadic infectious diseases, right? Once they're endemic, absolutely, we've got seasonal flu vaccines that people know how to address with capitalism. The second problem is a pop psychology problem, but humans are notoriously bad at planning and especially at dealing with probabilistic future events. And this is picking up on something Tillman just said. This is the second problem. So we know with certainty that there will be outbreaks and pandemics in the future. We just don't know exactly when, but we also know which pathogens are most likely to cause these. Most likely, not definite, not on a specific date, but we can prioritize and we can make some pretty highly educated guesses. But there's currently no commercial model that gets this work done. And so the sum of all of our push and pull incentives meant to cajole the free market led us to 2020, right? And so those are the problems. And I would assert that existential problems deserve multiple solutions, and the Pandemic Security Initiative is one solution that we're putting forward now. It's a public-private partnership model that's not just advocating for the work and not just funding the work, but actually doing the work to identify and then develop and then stockpile mitigations, whether these are vaccines or therapeutics or tests that we know are likely to be needed to prepare for future pandemics. And so we're working in collaboration with Dartmouth and other universities, CROs, CDMOs, biotech and pharma companies, and the federal government to actually prepare medicines and tests in anticipation of future pandemic threats. So as a nation or as a species, we have to do two different things here. And I think this is a really important distinction. We have to continue to invest in rapid response technologies. And so what Pfizer and BioNTech and Moderna and frankly, many of the other also RANs that are a half step behind, what they did in the last year was astounding. And that was enabled by investments in basic research, some of which went on at Dartmouth, as well as investments in powerful platform technologies. And Adimab's antibody discovery platform is certainly one of those. Great example. This is crucially important, but we also have to prepare, because rapid response is never rapid enough, right? And so Tillman's description of having a broad spectrum intervention that is future-proof to some degree against diseases that are not yet mutated but are likely to emerge is a great example of actual preparedness. This is preparedness versus responsiveness. And so identifying, developing, and stockpiling medicines so that clinicians can actually take something off the shelf on day one, and patients can be treated on day one, potentially even immunized on day one. These are possibilities. It's just a question of will. And so in 2020, we collectively declared war on COVID. Then we all sat down, we started designing our fighter planes, right? And this is not a good way to wage war. And so even though our industry smashed speed records developing vaccines and while maintaining very high quality standards, we responded exceptionally well. And this is what that feels like. This is what an excellent response feels like. It's us talking here on Zoom rather than being in person. It's all the disruption we felt. That's an excellent response, an outstanding one. And so responding is simply not enough. We need to really focus on, invest in a broad suite of preparedness options. It's really not an option, it's a requirement. And that's what we're trying to do through the Pandemic Security Initiative. I'll stop there and I'll look forward to questions in the Q&A.
Well, great. Well, thanks to all three of you again for making these fundamental points that yes, we did get to vaccine very quickly, relatively speaking, but we did that because of more than a decade of investment that pre-existed this pandemic. And as you just said, Jake, no matter what we did, it wasn't enough and it wasn't fast enough. We see that in the terms of the toll that the pandemic has taken, and we've got to be better prepared for next time. And as you said, the private market itself is not going to be able to do that. It is going to take a level of societal investment of governments around the world joining with the private sector to create the capacity to keep the science going and be ready for the next one. So we'll go to some questions now from the audience. A lot of them do bear on the fundamental science as opposed to the translation of the science. So let's take up a few of those science questions. Now, one questioner wants to know, what is our understanding of the current duration of immunity, including all the forms of immunity, whether it's antibody responses or cellular responses following effective vaccination? I'm not sure which of you might want to tackle that, but Kendall, I'm going to throw that one at you first.
Well, I think the data is being collected on that one. We just issued an emergency use authorization in January, so we're following up on, clinical trials recipients as we speak. So how long is immunity? How strong is immunity? Is it more, are you seeing more immunity, different kinds of immunity? These are ongoing questions. There's a lot of research to be done.
And just to add to that, one of the big challenges here when we're responding very rapidly is that long-term testing, these types of questions, they simply cannot be known until the time has passed, right? And so if you're doing it in a responsive mode, then we have to wait for these answers and really go on faith.
And of course, the situation is not a static one, with all of the emerging variants now, by some accounting, well over 1,000 variants circulating here in the United States. This story is going to be an unfolding one over time anyway, as we know. Let's ask just a related question. Do we know?
Can I just add something to that, Susan, just on that question? Because I think it's an important question. The durability of the response, we just started vaccinating people, so who knows? I think there are two data points that we need to consider in this context, though. There are other endemic beta coronaviruses that have been studied quite extensively prior to the emergence of SARS-CoV-2. And what we know from those viruses, like OC43, immunity lasts for 6 months, and then the probability of reinfection increases dramatically. So we don't know whether that's going to be the case here or not, but we have studied other coronaviruses, and that's what we're seeing there because they've been around for longer. Data point number one. Data point number two is a follow-up question. When you say immunity against what? Immunity against the identical virus or the variant? And there also we're starting to see data. We had this case that was published very recently about the town of Manaus in Brazil, where you had essentially 75% of the population had SARS-CoV-2 early on and essentially approached herd immunity. And then when the new variant showed up, all of a sudden hospitalizations and cases are skyrocketing again. So that's within 9 months, the new variant was able to outsmart the old immune response. And so that's what we know. And we'll learn a lot more every week that goes by, but it's certainly concerning.
And of course, that does suggest that we're probably going to be in an environment where booster shots for COVID-19 vaccines are going to be necessary over time and adapted potentially to the various variants, as is the case, obviously, with the annual flu vaccine. I do just want to quickly pull up another question related to the one we just addressed. This speaks to, though, the type of the vaccine. Do we have any evidence that duration of immunity differs at this point among the vaccines, the mRNA vaccine versus adenovirus vaccines, such as Oxford, J&J's, et cetera? I think the answer is we don't have that evidence yet. But again, quickly, Kendall, response to that one?
Yeah, no, you're right, Susan. These are important questions that we're going to be asking over the next couple of months, but the data is still coming.
Okay.
But for example, we know on the South African, there's 2 recent South African studies on the Novavax and the J&J vaccine. Efficacy against the new South African variant drops dramatically. And so, again, it's not just a question of how long, but how long against what. We think of this as one disease, COVID-19, in one virus that we call SARS-CoV-2, that's not the world we're in. We are now dealing with multiple variants, and while it may be in the thousands genetically, the relevance is really a subset of perhaps 5 right now, but it's that are relevant, that are able to escape the immune response, and that has to be, I think, carefully monitored, and we don't fully understand that yet, is my view. Kendall, you agree? Or?
I totally agree. In fact, it's going to put a tremendous strain on the current system that we have for genetic surveillance, both globally and domestically, right? Currently, I think we are sequencing 0.5% of all of the positive samples that we have. And estimates are it needs to be at least 5 to 10%, just to be able to have a snapshot of what we have where. And then we need contact tracing to follow up to see what are the effects of these new variants. And we need an entirely new laboratory infrastructure to test, to create neutralization assays to test, are the current vaccines effective against these new variants? And we need to standardize these so we can compare the results from multiple labs across the country and across the world, which is going to be a whole new level of coordination for everyone. And then we're going to be in a world where hopefully, actually, it's going to be really hard to have a naive population, to have a control group that has not been vaccinated, right? So how are you going to run your clinical trials? And we're going to need to establish correlates of immunity so we can start to run clinical trials to see if these new booster shots are in fact effective against the variants of concern. And it's going to be regional. We're going to need, this booster shot in this region and this booster shot in this region, and there's going to be mixing and matching, and it is going to be complicated.
I think this probably underscores a really important point that we want to leave the audience with, which is this: despite all of the concern and legitimate question asking on the part of the public about the safety of the vaccines, we're really in a world now where we could be asking, are the vaccines, not just are they going to be safe, are they going to be effective enough given what we've just been talking about, the spread of the variants, et cetera. So there's a lag, I think, now between the public understanding of where we are and where the science is actually pointing us, which is to some real uncertain places.
I would just add to that, Susan, I think that's a very important observation. But when you look at even the reduced efficacy, the severe outcomes are much, much lower. So let's not get too excited. Mortality in the vaccine trials has been dramatically reduced in all of them, even if the efficacy is reduced, defined by certain clinical criteria. So we are without question in a better world, and anyone who can get a vaccine should get it.
Yeah, can I put this is a really important point. Let me add just a little bit more color to it. So the way that the efficacy is measured is by infection. It's not really by clinical outcome after infection, right? And so if someone is infected, then it's considered a fail. Whereas to that person, if they get infected and they have some mild symptoms, that's a win, right? And so this is something to really be careful about. The other thing I'd add to this, we've got a lot of scientists who get right into the primary literature and the details of the vaccines. There are things that we can do from a public health perspective that are effective against all the variants, right? Stay home and work from home if you can. Wear masks, wear them properly. Distance, all that stuff that everybody's bored of hearing about and everybody wants to stop doing is effective against every single variant. Now, maybe a little bit different depending on transmissibility, but this is a way that we just need to continue doing what we have figured out how to do over the last year.
Great points, all of you. Thank you. So we have another basic science question, which is about the mRNA vaccine. How is it actually made? Where does the messenger RNA come from? This questioner wants to know, is it naturally occurring messenger RNA? Is it synthesized mRNA, et cetera? I have concerns about the effect on the body. So Tillman, let's throw that one to you if we could.
It's synthetic. It is made by synthetic organic chemistry means, and then it is packaged in a liposomal particle, and then it is delivered to the body. And when it gets into a cell, that cell is now reading the instruction of what the mRNA tells it to make. And the mRNA, in the case of a vaccine, tells the body cell to make a form of the virus, just the receptor binding domain of or the spike protein of the virus. So the human body now makes the spike protein. The immune system says, I have not seen you before, and starts mounting an immune response. And that is what then ultimately leads to protection. So it's new. There were concerns at the beginning. Early clinical trials at high doses had some adverse events, like fainting and muscle aches and things of that sort. That's still the case. But again, I personally would consider those to be safe vaccines. We've now dosed millions of people, and safety is not a concern from my perspective.
I heard a hint of concern about immunogenicity in that question. And so I think maybe it's important to state that, you know, these, at least from the Pfizer vaccine, are being made in E. coli, and then the plasmids are clipped and linearized, then they're transferred from that DNA into RNA. And so from the human immune system, there should be nothing that should be immunogenic in them. That's the expectation, at least. And that's been the observation to date.
Great. Okay, we'll keep going. We have quite a number of questions here. Another questioner wants to know, and this goes back to comments that Tillman made, if the coronaviruses are recombining now in bats, does it even matter if we can, through vaccination, get to stop it recombining in humans? I think you want to address whether we do stop it recombining in humans with a vaccine first, but then address the broader question of what is going on in the zoonotic population of animals as these viruses continue to recombine, Tillman.
Yeah, so there are two really separate events. The first one is there's genetic diversity created in animals. In this genetic diversity leads to new forms of the virus that on occasion can spill over into humans. And just because they spill over doesn't mean it's going to cause a pandemic. And as I said before, we can see in southern China that people that live in rural China, about 1 to 3%, have been exposed to coronaviruses most likely from bats. They don't know that doesn't mean there's a pandemic there. So there are specific events that have to occur randomly, more or less, that then lead to a pandemic. And what we've seen is two major outbreaks. The SARS outbreak 2003, 2004, and this one now, from this family of viruses that are present in bats. So now we're in a completely new situation. Now it has made it into humans. We have clearly human-to-human transmission very effectively. And the human immune system is now pressuring the virus to respond to that. And that is what we're now seeing, and that is what we're now starting to understand. And it is becoming pretty clear that it is the initial immune response, which appears to be a response that is very common, because if you look at the antibody repertoire, it's very similar. Whether it's us, people in South Africa, they're all very similar. And the virus now is seeing that pressure across many, many different people and is able to evade around that. And that is leading to these new variants. So I'm not sure if I'm answering the question. All I can say is fewer viruses is a good thing, because the more you have, whether it's in bats or in people, they have an opportunity of learning something to be more transmissive, and we don't want that.
Okay.
So just to add a very small amount to that, you know, bats are exceptionally dirty creatures. They have different immune systems and carry an awful lot of viruses around with them. That's the bad news. The good news is that there really isn't genetic selection pressure for transmission zoonotic transmission between the bat to the human. That's just a probabilistic effect. Darwin isn't helping that one along. And so it's one of these things that will happen and will increase when contact with these animals and humans increases.
So we have a question that speaks to the translational aspect of basic science and moving into the trial arena. This questioner wants to know, would it be possible to to hasten the testing process and essentially open up the phase 2 approval process by making it more of an open trial, so anybody could volunteer to get the vaccine? So we wouldn't have the controlled phase 3 trials, apparently, as this questioner wants to know. We would essentially open it up in phase 2 and throw it open to basically a non-controlled trial. Could we do that, and would that be wise? Jake, I'm going to throw that one to you.
Sure. So, I mean, this is an ethics question more than a regulatory one, really. FDA has rules about how things get approved, including emergency use authorizations. But FDA has also been very flexible and has really understood that what they are managing is not just risk and not just benefit, but risk benefit, right? The ratio of these things. And so, there are many proposals out there for how to speed trials, how to sufficiently inform participants in trials such that we could make things available earlier. There have even been proposals that for certain diseases that we simply don't have trials. And as long as people are making an informed consent to participate, then we do basically a phase 4 from day one. And so that's one end of the spectrum, right? But there are ethicists and regulatory folks that are constantly looking at these possibilities. It does speed up the cycle time, but if we have a coronavirus that is able to continue in its human hosts, mutating and evolving essentially at a pace that is faster than we're able to get our approvals done and our new vaccines made, then we've got a real problem, right? And so it's really a question of can we limit the spread? Can we limit the number of people that are acting as reservoirs and the number of actual viruses that are under mutational pressure? The more of those we have, the more infected people we have, the more mutations we'll have, and then eventually some of them will gain fitness.
Great. So we have a question that has come in from someone who identifies herself as a physician in a correctional facility. They've begun vaccinating the staff and incarcerated patients. Several individuals tested positive after they got the vaccine. That's affected the buy-in of the population about their willingness to be vaccinated. How can she address that situation? Kendall?
That's a really tricky one. Usually, nobody wants a public health scold, right? So there should be no shaking of the finger, but there should be, engagement and education that, this is how vaccines work. First of all, even a 95% effective vaccine is not going to work 100% of the time. Also, it takes time to build immunity. So it's only perhaps 50% effective after the first shot. And it won't approach 95% until several weeks after the second shot, which is, does not impugn the vaccine itself. It's just how it works. And so, truly anyone is able to hear that message if they're approached with respect, in a way that they can hear the message. It doesn't do any good to, scold people or just throw information at them. There needs to be an engagement.
So Tillman made a point earlier that's highly relevant here, which is that those people who were infected probably had less severe disease, right? And so they may have ended up dead had they not had that first shot. And I don't know what the timing was vis-a-vis the first shot, but the simplest message and the way I try to describe it to people is that you are not vaccinated until 2 weeks after your second shot. Right. It's not the moment the needle goes in your arm, right? Keep your mask on, keep distancing, even after you've had that 2 week period, because that's when you have that additional benefit. But as Kendall's saying, none of it is perfect. Right.
Right. So another questioner asks a very important question. What can we learn from the rapid response this past year? And I guess I would amend that saying both a rapid response, but also in a way, as you all have said, not fast enough, given the dimension of the problem, but given the response of the past year, what can we do now to incentivize improved collaboration among universities, academia, industry, and governments between pandemics to prepare for the forthcoming ones? And Jake, I'm going to start with you on this. You spoke to this as did you all, but let's start with you.
Sure. So the analogy that I like to make here is to, for example, oncology and innovations in that space. And so the National Institutes of Health provides billions of dollars of funding every year, primarily to universities and to academicians like Kendall, like Tillman. And in their academic labs, they do work and they create innovation. And in the case of NIAID, which is most relevant to infectious disease, that's about $6 billion a year. And that results in 2 granted patents every single day. And so the innovation engine is running, right? The research money is there. NIH is, over 40 billion a year now in budget. And then the problem is that's where it stops, right? And so I have friends in the tech transfer world who call up and say, Jake, I've got all these infectious disease patents. What do I do with them, right? They just cost me money. And the problem is what Kendall mentioned, what I mentioned, that there is not a commercial market for a sporadic infectious disease. And so that's exactly what we're trying to focus on here and simply say, you know what, instead of making a bunch of sort of fake incentives, either push or pull, let's just say that this is a societal problem. This is a governmental problem. Have the feds put in the financing. And then on the back end, as a taxpayer, there's no pricing power. And essentially it becomes a cost plus situation. And then it gets done, right? And so that's what we're proposing. There's a lot of other proposals out there. I really think we should be trying a handful of them because of the importance of the problem. But if people have a path and if people have the resources to do the work, this will work, right? And the issue right now is one of resources.
So just to follow up quickly, Jake, you're describing a situation where there would be front-end government funded, that is to say taxpayer funded research to develop these, but then we would limit the profits that would come to companies that commercialize those products when we needed them?
That's the model that we're proposing in this specific instance for this public-private partnership. And the funding for research, again, I think is quite healthy. And there's a ton of innovation out in the world. What's not there is the money to do the preclinical development, to do the clinical work, and to actually move it all the way through in order to get to an approved product.
So Tillman, you've been on the side of launching a lot of these companies that depend on profits to continue their work. Do you think this kind of an approach would fly? Should it?
I don't know. It's hard to say. If you look at the past, the appetite has been pretty low, but I think given that we've gone through this situation, I think it may change. I want to address your earlier question of what should we do differently. I mean, I think we actually did spectacularly well. If you look at the time it took from identifying this new pathogen and its involvement in this acute respiratory disease to having the first patient dosed with a vaccine, and that vaccine actually being very efficacious in less than a year, that is a remarkable accomplishment. So let's go back to, I think Kendall said at the beginning, or maybe you said it, Susan, at the beginning, right? So let's not forget that. Let's also understand we got lucky. There are thousands of infectious diseases where you just simply express a surface protein and you get no immunity. We were lucky that this worked in this case, and the investment in platforms and the understanding of coronaviruses paid off. So that's how I see the world. Now, when it comes to Jake's proposal, I think the problem is the following. When you look at coronaviruses, again, it's a very diverse family. There are thousands of different coronaviruses out there. But you can then start breaking it down, which ones are likely to be a pandemic threat. So they have to, for example, have a zoonotic reservoir. They have to be in animals like I described before that they can recombine. That is typically a requirement, check. Next, they have to be respiratory viruses, because that's when you can actually make a living as a pandemic organism. If it's blood-to-blood transmission, you're not going to get it on an airplane, you're not going to get it in a restaurant. So again, not high pandemic potential. So you can start applying rational filters of where the threats are going to come from. And then you're going to still, in the case of coronaviruses, end up with dozens. What I would propose is, and the technology exists now, and that's been something that's been developed in the last 5 years, that I can now neutralize an entire class of viruses, because we understand how these viruses get into human cells. And they all do it the same way. They all exploit this ACE2 receptor. If I can block that, all the coronaviruses are done, or at least the ones that target this particular human receptor. So I think there's a process of thinking about what are the high pandemic threats, break them down into the classes that sort of share entry mechanisms, and then attack them as classes, not as individuals. And I think if we do that, I think we can make a real dent and make a real change in terms of how well prepared you. And then at least you have something to treat early on. That gives us time with vaccine development. And so anyway, that's what I would advocate for.
This is exactly the proposal. And so we have to prioritize the threats based on the impact and the probability. And we also have to prioritize the mitigations. And so a broad spectrum would be preferred over something that's narrow spectrum and less likely to cover a number of different viral threats. And so exactly right. There's also great use for innovative immune modulatory drugs, control of inflammation, some of the secondary impacts that a lot of these viruses have.
So a related question to all this, you've described the construct that has to exist to serve in a way as a kind of early warning system. How does it come about? Who creates it? Where is it housed? This capability to do the kind of work that you described.
That's for me.
Yep, either of you. Yep.
Okay. Yeah. So, again, we're envisioning this as a public-private partnership with a big capital P on the partnership. So there is an enormous amount of capability in the federal government, at ASPR, at BARDA, at NIAID, at CDC, at FDA. There is an enormous amount of capability in our university systems. You're hearing some of it today. There are public policy organizations, lots of other places at WHO that are looking at these prioritization issues and are looking at probabilities of emergence that are linked into some of the surveillance groups that are out there as well. And so I think Kendall makes the point best. There needs to be global coordination from surveillance and prediction all the way through to the last mile, and then phase 4 type studies. And that's something that simply does not exist today. How does that get done inside one or many governments? That's beyond my pay grade, but I know that we can contribute to that question of putting some drugs, some vaccines, and some tests on the shelf so that they're ready, and ideally broad spectrum, ideally having the ability to mitigate the most likely threats to hit us in the future.
Great. Well, we are getting close to the end of our time. I do want to circle back and give Tillman an opportunity to address a question that I posed to him initially, which is that we've seen the enormous success with COVID vaccines. We've also seen several of the therapeutic antibodies approved, but it does appear that they're not being used as widely as they could be used in the pandemic. Why is that, and how do we address that part of the equation of making effective use of the countermeasures that we do develop? Tillman?
So let me just describe to you what has been proven in clinical trials these antibodies are able to do. They dramatically reduce hospitalization. So if you come and you treat early, the likelihood of you developing a severe form of the disease and die of the disease are dramatically reduced. That's the good news. The bad news is that the current versions of these drugs are administered intravenously. So you have to be admitted to the hospital to administer the drug. And the problem that we have now is people come in, they're diagnosed, they would benefit from the treatment, but the way the hospitals are set up, you have to sort of admit them to give them an IV treatment. But in that situation, you have people that are on their cancer chemotherapy in the same setting. You have to clear all them out to now treat someone for a potential COVID problem. And the system is just not set up to do that. That is why we decided to not develop an intravenous form of administration, but an intramuscular injection. So you come, you're high risk, you get a shot in the butt, and you go home. And by the time you sit in your car seat, that will have disseminated out from your butt and protect you and prevent bad outcomes. So it's the way the drug, again, because we were going so fast, you couldn't invest time in formulation and come up with formulations that you could use in an intramuscular setting. And that's one of the problems that the current forms of the drugs that were approved under EUAs are experiencing.
So that's another very important translational issue. We have to have enough time to figure out actually how to use these products as effectively as possible, and sometimes it won't be the first one out of off the mark. Okay.
And sorry, Susan, just one closing statement on that. The problem is the folks that came out of the gate early, like Lilly and Regeneron, The Lilly antibody, this is the antibody that some of you may know about, was the one that was used to treat, I think Chris Christie was one of the more well-known patients. That antibody is completely ineffective against the South African variant. It does nothing. And so these antibodies that were developed early are very narrow, and the virus is already mutating around them and making them obsolete. And so that's, again, why breadth is so important, because that prevents that from happening.
Well, all of you, thank you so much for your wonderful and very instructive comments today. You've more than made the point several fold here that the vaccines were developed quickly, other things were developed quickly, not because there wasn't substantial investment ahead of time. There was a great deal of investment ahead of time, particularly around developing the vaccines that have come to be used against COVID-19. So this has been a culmination of a long period of scientific discovery and development. As Kendall, I think, put it, 1,000 things had to go right to get these vaccines to market, and fortunately they have. And as Tillman said, we also got lucky to a certain degree. And what that all reminds us, as you all said, is that we've got to prepare for the next time around. These things don't happen naturally. I think, Jake, as you put it, rapid response is never enough. We have to be prepared, not just responding, as you said, in investing and developing and stockpiling these medicines. And it's going to take, as you all described, some very important public-private partnerships on the scale of which we didn't even achieve in this current pandemic, to be ready for the next time. As you all said, it's really a question of will. And therefore, to be better prepared, not only do we have to have these partnerships, but we have to have some kind of very intensive system to look broadly and determine which pathogens are likely to become pandemic, and put a lot of effort into not only discovering them, but looking to develop all the countermeasures against them. Finally, as you all have said, we do need to address better the final phases of this whole set of activities, getting shots into arms, et cetera. And that's a subject probably for yet another day. So I want to thank all of you panelists for sharing your expertise on this subject. We also want to thank the audience for engaging in this conversation. I'm sorry we could not get to more of your questions. If you haven't done so yet, though, please do sign up for the final session in this series. As I mentioned earlier, Eye on Equity, which will be held on February 22nd. Please also do not forget to complete the 2-minute survey you'll receive at the conclusion of the program. Your feedback really does help us improve future programming. So have a wonderful rest of your day wherever you are. Be well, stay safe. Thanks again for joining us.